Hello. Hello students. Today we will discuss regarding a new chapter and that is evaluation of crude drugs. Okay. And which is very important on the basis of exam point of view, as well as it is very important for the different competitive examinations. Even somebody preparing for the GPAT as well as some your your examination, pharmacist examinations or any drug research examination, you may expect some question from this section. Okay, so first we'll discuss about what is the meaning of evaluation of crude drugs. So evaluation drug means your confirmation of the quality. Okay, confirmation of the quality of certain drug by adopting suitable method is called as evaluation of crude drugs, because we know that we are getting the crude drug from nature and there is a probability chances of your adulteration either by intentionally or unintentionally, because some crude drug which is available in the market, they are adulterated, they may be found in adulterated form either intentionally or unintentionally. Okay, so to know the quality of the drug, to know the quality of the drug, the purity of the drug, and the identity of the drugs, each carried out by each can be happened it can carried out by the help of evaluation of crude drug. So the main objective of evaluation of crude drug to identify the drug, to determine the quality of the drug, to know the purity of the drugs. Okay, so now next we'll discuss about what are the reason behind that we need to evaluate the crude drug. First point is there is a changes in or variation in botanicals. Botanicals related to crude drugs which obtained from markets, which obtained in the market. So there is a changes in botanicals. माने कुछ ड्रग होता है, ओके, जिसका जिसका वेरिएशन, केमिकल वेरिएशन चेंजेस होता है. आप जानते हैं कि एक ड्रग, द प्लांट कंटेन द सेकेंडरी मेटाबोलाइट्स. ओके, द सेकेंडरी मेटाबोलाइट इज वेरी वेरी इंपॉर्टेंट फॉर अस because the secondary metabolite we are used as therapeutic effect for the as a drug, because it having therapeutic action or pharmacological actions. But you know that same two plants, if cultivated in different condition, then there is a chances in variation in chemical constitutions. That means suppose A is a plant, another plant. If a plant is cultivated in one geographical location and same A plant is cultivated or found in another geographical location, then it is not mandatory that the chemical constituent of the A is same like the chemical constituent of the another A which is located in different location, because due to the climatic variation the chemical constituent concentration or secondary metabolic concentration vary from place to place of same species. So that is the reason there is a changes or variation of the botanicals of the same plant, and that can be identified by evaluating the crude drugs. So before incorporating any crude drug for the formulation, it's need to identify, it need to evaluate, it need to standardize before any formulation preparations. Second important thing that to determine the quality control of the crude drug, because evaluation of a crude drug that help you to know the quality of the drugs, the period, the quality of the drug, whether it is good quality or bad quality, that can be known by the evaluation of the crude drug. Third one is that identification of common adulterants. You know that adulterants are the substances that are added partially or wholly to the genuine substances to bring more profit. Okay, so those substances added to the genuine drug, they are called adulterants. So due to addition of the adulterant, the drug quality is became substandard or deteriorated. So that can be identified by evaluation of the crude drugs. Fourth important point, your safety assessment or documentation for toxicological activity. So here I want to say something that no drug, it is people have idea that generally plant derived drug or animal derived drug or natural drug are safety, but that is not 100% right, because there are so many drugs available which are derived from plant sources, animal sources, or any other nature sources. They have also side effect, toxic effect, or undesirable effect. Okay, so that is the reason it is not right to say that the all the derived natural drugs are safety. No, drugs are there. Okay, they have also they have also toxicological activity. So the safety assessment can be achieved through the evaluation of crude drugs. The same safety assessment can be documented for the future use. Okay, that's why the toxic activity like LD50, ED50 active, like your tolerance activity, everything should be uh, kept in a documented form, so that that can be assessed in the future to assess any type of drugs. Okay, next slide we discuss about that what are the methods available which is useful, in what are the ways in which one can evaluate the crude drugs, because evaluation is the most important thing. But how the procedure, what are the procedure, how you do? Now, there are so many procedures, but processes are available, like first one, your morphological identification, or it is also called as organoleptic evaluation, or it is also called as macroscopic evaluation. Macroscopic evaluations, that means these all morphological, organoleptical, macroscopical, all are the same. Okay, so here in the first case we are evaluating the crude drug on the basis of morphological differences, on the basis of morphological study, or on the basis of your organoleptical study. I will discuss in the complete when we come to the individual evaluations. Second one, most important, the microscopical evaluations. What does it mean? You know that the different crude drugs are there. They have different type of microscopical tissue arrangement or cell arrangement, or microscopical some substance may be present. So the microscopical character that guide us to identify a particular drugs. The microscopical character that help us to identify different species of same plants. Okay, so in this case we are using a microscope, either in the compound microscope or light microscope or simple microscope, which guide us to evaluate the crude drug on the basis of microscopical study. We will discuss elaborately on the when we study all this point. Third one is your physical evaluations, is the most important one. So physical evaluation contain so many parameters. Many parameters are there, like your refractive, like your extractive value, as value, moisture content, flow property, viscosity. Many are there. Evaluation. It is not a single one. Physical evaluation consists of series of evaluations. So that also sometimes help you to know or to evaluate the crude drugs. Okay, and fourth one is your chemical evaluations. What does it mean? The chemical evaluation is a procedure, very common procedure that is employed, that is used to evaluate the drug on the basis of their chemical constituent present in it. We know that two type of, what that means, the chemicals which present in the crude drug are categorized into two categories. One is called primary metabolite, primary metabolite. Second one, your secondary metabolites. Okay, primary metabolite are those chemical or those biomolecule that is very important to the plant itself, because plant grow, reproduction, all the physiological activity that is occur in a presence of primary metabolites. Whereas the secondary metabolite are a group of chemicals that derive from the primary metabolite by a certain pathway that is called as secondary, that is known as popular known as secondary metabolic pathway. Okay, to produce secondary metabolites. The secondary metabolite is important for the plant as well as it is very very important to the animals or the human being because we use the drug. Why? Because the crude drug contains the secondary metabolite which is our interest. So this secondary metabolite can be identified by adding certain chemicals, and the observation guide us to know what type of chemical constituent present in the crude drug. That is called chemical evaluations. Chemical evaluation again two type. Okay, one is called your qualitative evaluation. Second one is called quantitative evaluations. Well, qualitative evaluation means we can know the what type of chemical constituent, what type of secondary metabolite present in the plant. That is called qualitative evaluations. Qualitative chemical evaluations, which also some people are saying it's known as preliminary phytochemical screening. The technique we are using to identify what type of chemical present in the plants. Type of chemicals means like glycoside, alkaloid, tannin, volatile oil, fixed oil, proteins, carbohydrates, okay, gums, mucilage, terpenoid. These all are the group of secondary metabolites, whether present or not. That can be done by qualitative chemical evaluations. Whereas the quantitative evaluation that says about how much the amount of the desired chemical present. That is quantitative chemical evaluations. So apart from 1, 2, 3, 4, another one that is known as your it is called your biological evaluations. Biological evaluation. Actually, in this video today I will only say about introduction part. On the next video we will discuss about individual type of evaluation. So biological evaluation is one of the important tool that is useful to evaluate the crude drug on the basis of their biological activity. Okay, biological activity means the crude drugs activity on the biological body. That is called biological activity. Okay, whenever some drugs which cannot be evaluated by morphologically, microscopically or chemically, then finally we can depend upon another evaluation that is called biological evaluations. Biological evaluation means we can evaluate the crude drug, you can evaluate the potency of the crude drug either on the microorganism or like fungi, bacteria, or in the animal body or animal tissue or whole animal, like that ones. Biological evaluation is also called as bioassay. Okay, so many type of bioassays are available that is useful to identify the purity or the quality or the toxicity of the drugs. Okay, so this biological evaluation also help you to do the test on the human being or healthy volunteers. So when somebody evaluate the crude drug on applying in the biological body, particularly living animals like rat, pigeon, your mice, like your rabbits, etc., they are popularly called as bioassay. It is, and this condition, this phase is called as preclinical trials. So whenever a drug, maybe synthetic sources, maybe natural drug, is successfully passed in the preclinical trial, then it will move to the clinical trial. Clinical trial means here we are actually identify the drug by applying on the human volunteer. Okay, but again it is 4 groups like phase 1, phase 2, phase 3, 4, like that ones we will discuss later. So this is about your introduction video of evaluation of crude drugs. Very short video. So maybe question arises, very short question. Now, what is the definition evaluations of the crude drug? Why it is very important to evaluate the crude drug? And maybe you can get the question, what are the different methods of evaluation of crude drugs? Okay, on the next video we will discuss elaborately of individual evaluation, like we have to study regarding morphological evaluation, microscopic evaluation, etc. Thank you very much for your patience, for the kind listening. Have a nice day.